Good afternoon. Okay. So my name is Chris Polson. I'm a professor in Earth and Environmental Sciences and also the Associate Dean for Natural Sciences. And I have the great pleasure of introducing the fourth of our four UMBS director candidates. Before I introduce Cori, I want to thank EEB, staff in EEB, Bali, Nancy, Carrie, Dearman for hosting the visits and organizing the visits. We couldn't have done it without you, so thank you very much. So Cori, does she need an introduction? She got her bachelor's degree at in biology and in chemical engineering, and then she got her PhD from this department in 2007. She then did a postdoc. She started her academic career at Tulane University, and then now she is at the University of Pittsburgh. She's an associate professor there, and she's director of the Hymetumi Biological Station, Lab of Ecology, which has a lot of parallels with UMBS. I'm not an ecologist, but her work is fascinating. If you read the titles of her talk, first of all, they're very understandable titles, so congratulations on that. A lot of her work looks at how amphibians and reptiles are affected by fungal and how that is affected by climate change. And so I'm really interested in that because I'm a climate ecologist, so I'm very much looking forward to Cori's talk. So without further ado, hand over the stage to Cori.
Sounds good. It's a real pleasure to be here today. Can everyone hear me okay? Okay, great. And it's a strange and wonderful pleasure to be back at one's alma mater under these circumstances. And so I'm really excited to tell you about some of the research, one part of the research that I've been up to since I left here 12 years ago, and also conclude with some thoughts about my vision for the UMBS. Throughout this talk, you'll be seeing some wonderful artworks, which I just wanted to mention briefly are the result of an artist in residence who I had in my lab for a semester, which was an amazing experience, and I like to show off her products whenever I can. I'm going to start by just acknowledging that the work I'm about to talk about was a product of many, many, many people's hard work, lots of collaborators, a large number of lab members, work at several different field stations, and funding from a variety of different funding agencies. So keep that in mind as we go through. So I'm here today because amphibians are amazing. I put this slide up here and made this slide to represent some of the amazing amphibians I've had the pleasure to come to know working in Panama. And the thing that originally attracted me to amphibians was their diversity, not only in form, but in reproductive mode. And so some of those things that are shown up here, you can see this guy who's got eggs in a pouch on his back. You can see that one up there who's carrying tadpoles around on its back. And that father who's guarding egg clutches that were probably laid by 3 different females because they seem to be at different stages of development. You can see the amazing diversity of form, brightly colored toxic aposematic species and species that are super good at camouflage. And so when I was starting grad school, one of the things that motivated me to go into biology was the news that was coming out that was talking about amphibians using words that I never wanted to hear, things like mass extinction, biodiversity crisis. And so at the time when I was starting grad school, we were just starting to figure out what were the main drivers of this crisis for amphibians. And so the data we have that talks about the threat to amphibians globally is a little bit out of date now. The last IUCN Global Amphibian Assessment was done in 2004. They actually have done a new one, but the data are not all out yet. But at least with these old data, you can see that 30% of all amphibian species are listed as threatened. Another 25% are data deficient, meaning we don't even have enough information to put them in a threat category. And sadly, only 17% of species, we can say that there's no real threat perceived. So there's a lot of room for conservation and a lot of room for thinking about how our activities on this planet are impacting amphibians. When we look at the major threats to amphibians, you see a lot of things that we would associate with major threats to many different taxa, including habitat loss, pollution, invasive species. But on the list of top 5 for amphibians is disease. And we used to think that that was somewhat unique to amphibians, but it turns out that there's a lot of emerging pathogens that are threatening wildlife. And I've put up here, I've highlighted fungal pathogens because these are a major threat to mainly ectotherms, but sometimes endotherms when they behave like ectotherms, like hibernating bats. And they oftentimes, we find links between these emerging fungal pathogens and climate. So I'm going to highlight that as we go through. So to tell you a little bit about the fungus that I study, it is a fungus, it's actually 2 fungi now in the genus Batrachochytrium, part of the chytrid group of fungi, and they cause a disease called Chytridiomycosis. I like this picture because it's beautiful, but some of the information on it I wouldn't necessarily call completely correct. So the species that I'm going to talk to you about today is Batrachochytrium dendrobatidis, which we like to call BD, so that we don't have to keep saying that over and over again. And this is a species that we now know infects over 500 species of amphibians worldwide. There's another species that was more recently discovered called Batrachochytrium salamandravorans, or B-sal for short, which now is infecting and causing declines in European salamanders, but has yet to make it to the United States that we know of. So for Chytridiomycosis, I was part of a recent study that sort of tried to look at the global impact of this disease on amphibians. And one thing I wanted to highlight is we learned about what this pathogen was right here in the late 1990s. But by the time we knew what it was, the impact that it had had on biodiversity for amphibians was already well underway. So here we see extinctions that are related to Chytridiomycosis, presumed extinctions, and different levels of severity of decline, and how in some cases those declines are continuing to happen even to this day. This figure up on the right shows sort of the taxonomic distribution of these declines due to Chytridiomycosis. And in some cases, for some taxa, there doesn't seem to be a lot of signal, but for others, like this genus Atelopus, you see a lot of species that are either presumed extinct or have had major 90% declines due to this disease. The other thing I wanted to point out is that this disease is global, but its impacts have been more impactful in some parts of the world than in others. So in Central America, we see more species in the categories where you don't want to find yourself. Also in South America, and it doesn't bear out in terms of numbers, but there's been big effects in Oceania as well. In North America, the pathogen is present and prevalent, and we have had extinctions and declines, but it doesn't seem to be having the same level of impact as it has had in other parts of the world. So I'm going to tell you about a story of the Panamanian epidemic and my work that we've done there, and then I'm going to end with some work that we've been doing in North America that relates to climate and disease risk. So I started my PhD in 2002 here in this department, and my research took place in central Panama. And so my research took place in El Cope and El Valle from 2002 to 2007. So I was right in the center of the epidemic of Chytridiomycosis as it spread through Panama, which was not my plan, but it turned out to be the reality. So to give you a sense of the scale of what was going on when this epizootic hit Panama, this is a figure from a paper by Karen Lips and colleagues that shows the number of amphibian captures per meter per person. So it's sort of a proxy for density along these riparian transects over time. So we have these nice, healthy amphibian communities. They're finding more and more frogs, more and more years they sample, and then the fungus arrives and the community crashes. And they start to find dead frogs along these stream transects. And so one of the projects that we've been working on, teaming up with people that I met as grad students in Panama, we were all sort of in the thick of this epidemic all at the same time, is what happens next? So even several years ago, it was assumed that this was the end of the story. These communities were gone and life was forever changed in Panama. But we wondered what happened. So we know that we had a naive host community. An epizootic event happened with many species becoming infected with this pathogen and many species experiencing declines and even extinctions. But what happens next? So it could be, what we saw during the epizootic was we started with high numbers of hosts, and as we saw the prevalence of the pathogen increase, the numbers of hosts went down. So what happens over time? Does it end with extinction of the hosts followed by extinction of the pathogen? Or do we see some sort of recovery in the host populations? And if we do, what does that look like? So we predicted that there would be, optimistically, a transition between this epizootic phase and an enzootic phase where the host and the pathogen could persist in the shared environment. But we didn't know what that would look like in terms of the pathogen and the communities of hosts. And so we had funding from NSF that basically aimed to answer these three questions. How does the community composition change over time? How do BD infection patterns shift after the epidemic? And how do host and pathogen come to coexist? So I'm going to talk about the first two questions first and how we tackled those. So in 2012, we started monitoring 15 different sites that I had sampled from my PhD and doing visual encounter surveys for any amphibian along these riparian transects that we could find. We recorded anything we thought could potentially be important to understanding whether species are coming back or not. And critically, we took diagnostic swabs, skin swabs, to test for the presence of the pathogen using quantitative PCR. Another thing that we did was critical was when we found dead or sick frogs, isolating the BD from these frogs so that we could have new isolates that we could work with and compare to isolates that had been made during the epidemic. So how did the community change? So I'm going to start by illustrating with just this one species, the little cute little Panamanian rocket frog, which was very common in El Copay, the region with the star here, prior to the arrival of BD. Once BD arrived, we did not see the species for several years. We didn't see them and we didn't hear them. They're a very vocal species, so you hear them before you see them usually. When we went back for the first time in 2012, we didn't find them, but in 2013 and 2014, we did. And in every year we've gone back since then, we've found more of them in more places and in higher densities. We also found in our first time back in 2012, Atelopus varius, which is a species that was considering being listed for extinction at the time when we went back to look for it. So we were really pleased to see this first evidence that some of these frogs were recovering. When we look at this at the scale of a whole community of amphibians, we can compare the communities prior to the arrival of the pathogen at our three different research sites, research areas. And so that's what these ones are on the left-hand side. And you see in each case, when the pathogen arrived, the community changed. But we're happy to see that in each of these cases, after that change in the community, we're seeing a gradual sort of return to the community that was there before. And that was not something that people had been expecting to see. And we can also see that in terms of densities of particular species along our transects. So when we compare transects in terms of numbers of individuals found of several of our species before the disease arrived, during the epizootic, and now during what we think is the enzootic phase, we're finding that many of these species are returning to their original abundances, which is a really good sign. And you may be asking, okay, are they doing this because the pathogen is gone now? No, the pathogen is still there. The pathogen is there. It is less prevalent, meaning that it affects a smaller proportion of the amphibian community than it did, but it is still present in all of our field sites at every one of the streams that we sample. But it is only affecting about 20% of the amphibians, whereas during the epizootic phase, it was impacting a greater number of individuals. So the amphibians are persisting with the pathogen in what we think is now the enzootic phase of the disease, and we've found that the prevalence of the pathogen is lower. How are they doing that? That's the next big question. What has changed? How have the pathogen and the host come to coexist? And for this, we had two competing hypotheses. We figured perhaps the pathogen has shifted in pathogenicity, become less pathogenic. Or maybe the hosts have become better at defending themselves against the pathogen. And I have to say that our money was always on the pathogenicity part, because pathogens just are known to evolve faster than their hosts. In order to ask these questions, we relied upon a collection of historical BD isolates that were made during the epizootic. So we had a set of isolates that were taken from dying amphibians during the original declines. And we had our contemporary isolates that we isolated about a decade later from amphibians that had been coexisting with the fungus for that decade. And so we looked at this question of pathogenicity in a couple of different ways. We looked at it from the standpoint of the sort of the physiology and life cycle of the pathogen by comparing things we thought would be related to pathogenicity, including growth, which we measured with changes in optical density in an aquatic solution. We measured the density of zoospores, which is the reproductive motile units that infect the skin of the amphibians, and how many of those are produced per life cycle. And we also looked at zoosporangium size. The zoosporangium is sort of an urn-shaped container that contains the developing zoospores. And the larger the size of those, the more zoospores they can contain. And so that affects potentially how many individuals can get infected per generation. So we compared those things. And in any of our comparisons that we did not find any difference in growth rate, zoospore production, or zoosporangium size when we compared our contemporary isolates to the historic ones. So it did not seem like any of these aspects of the fungus had changed. The other way we tested for differences in pathogenicity was using assays that took the host into account. So one thing we know about the BD pathogen is it has an ability to inhibit the immune systems of frogs, and it can particularly inhibit the proliferation of lymphocytes in frogs. The other thing that we did is we exposed a susceptible species to the pathogen and exposed them to either current or historic isolates of the pathogen to see whether one had more mortality than the other. Okay, so when we looked at the lymphocytes, we compared the historic and the contemporary isolates, and we found that they each inhibited the proliferation of amphibian lymphocytes to some extent, but there was no difference in that inhibition between those two categories. So they were very similar in their capacities to evade the amphibian immune defenses. When we looked at pathogenicity in a susceptible host, we exposed, these were frogs that were extras from captive breeding programs in zoos, either Atelopus various frogs that were known to be highly susceptible. We exposed them to either historic or contemporary isolates and looked at infection intensity and survivorship. And in neither case did we see any difference between the historic and contemporary isolates. Both historic and contemporary isolates led to 100% mortality by 40 days in this species. So they were all very highly virulent. So that leads me to the next question of whether hosts have evolved better defenses. And we focused this on defenses that are found in the mucus on amphibian skin, because this is a skin disease, and the most important defenses we think are on the skin. So amphibians secrete in their mucus antimicrobial peptides that are made in the granular glands in the skin and secreted into the mucus. And so one of the things we can do is look at those antimicrobial peptides and how effective they are against the BD pathogen. So we were lucky in that people had made collections of these antimicrobial peptides prior to declines in a number of species from Panama. So we had a good sense of how effective these peptides were inhibiting the growth of BD. And so when we went back to these same species and collected their peptides a decade later, once they've been living with the pathogen for a decade, we were able to compare, and what we saw was that in a number of these species, they have peptides that are now more effective against the pathogen than they were a decade ago. Another thing we did was we compared the peptides between Atelopus, now this is Atelopus, various individuals that had either been living in the wild. Some of our extant populations have been living in the wild with BD for the past 10 years, or those that had been taken out of the wild and bred in captivity. They were taken out of the wild before BD arrived, so they were completely naive to the BD pathogen. The captive frogs were not able to inhibit the growth of BD, whereas the ones who had been living in the BD community for a long time had really effective peptides. So to this suggests to us that the evolution of better host defenses may be a key to surviving in an enzootic state with the BD pathogen. And one of the things that we're working on now is trying to understand sort of the mechanisms by which that's occurring. So we have a handful of sites where we have data, we have genetic samples from Atelopus from my dissertation research from way back when, before the declines, and we have genetic samples from those same places now. And we're using whole exome sequencing to test for selection on genes that might be involved in the defense against Chytridiomycosis. And that work is not done. It's being done in collaboration with Bree Rosenroom and her student Allie Byrne. And Allie's finishing up this project very soon. But the cool thing is we're seeing some really interesting things pop out, like genes that are involved in antimicrobial peptide production. We're also continuing to monitor these communities, and we're coming up with more and more clever ways to find these needles in the haystack, including using environmental DNA sampling, collecting water samples and using environmental DNA to figure out what frogs are using those waters. We're using acoustic recorders to try to detect things that may be out there and calling in times when we're not out in the field. And we're working with our local communities to have them contribute to helping us to monitor some of the populations that are at risk. Okay, so that's our work in Panama, and now I'm going to transition a little bit to thinking about this disease in the context of climate. And so as I do that, I'm going to think about this sort of important interaction between host and pathogen and environment, and how that goes forth. And so when I think about this, I like to come back to the eco-health concept, which says that the health of all species and their environment are inextricably linked. And we can see evidence for that in terms of the annual cycles of many human infections. And also when we think about some of these fungal pathogens of wildlife that I mentioned before that have clear connections to certain types of climate or certain environmental conditions. So the work that we've been doing in this context is attempting to answer the question of how current and predicted future changes in climate influence disease dynamics and how host immune systems function. And the work that we've been doing on this has been happening across the U.S. The stars in orange represent field stations where we've done our work, and the other sites are a range of other wild areas where we've done our work, and just put out these pictures to show the community of amphibians that we've been working with here in the U.S. Okay, so for some background, there's a lot of reasons to suspect that temperature is really important to the interaction with BD. So in culture, BD grows best around 23 degrees Celsius, and depending on the isolate, dies somewhere between 28 and 30 degrees Celsius. So it can't tolerate very high temperatures very well. And when I was doing research in Louisiana and northern cricket frogs, it seemed like our very seasonal prevalence that we saw in this species, which is the dark black line, could easily be explained by air temperature. So air temperature, high air temperatures are these gray lines, and every time the high air temperature got above where BD could tolerate, the prevalence in the population would go down, and we'd see that repeatedly. So this was suggestive that there was a really clear relationship between temperature and disease dynamics in these populations. And so our work across the US now has really borne that out, in that we see these sort of seasonal cycles in both the prevalence of BD in amphibian communities, and we see it also in terms of how infected animals are. The intensity of infection is very seasonally prevalent, whereas the heaviest infections are usually in the coolest time of the year, and amphibians have lowest infections in the warmest time of the year. But when you're doing these in a field site, it's hard to really isolate what is the causal factor. Is it temperature? Is it precipitation? What is it about the environment that's causing these seasonal cycles? And so one of the ways we've tackled that is using exposure experiments and really isolating the role of temperature. So my graduate student, Julia Sun, did this with northern cricket frogs. We did an exposure experiment where we exposed frogs to BD and held them at a variety of different temperatures. And what we found was that we had much higher mortality or lower survival at low temperatures, and as temperature increased, survival with BD infection increased, such that between 20 and 26 degrees Celsius, frogs were doing fine, but below that, there was a lot of mortality. And so this was interesting to us because based off of the thermal curve for the pathogen alone, 23 is the best temperature for the pathogen to grow in culture. But 23 was, when we did this in the frog, the temperature where we saw the least mortality in the frog. So it suggests that without considering the host, these sort of thermal predictions that we might make just based on the pathogen's physiology are not going to make the best predictions. So you can't predict the effect of the environment on the disease without considering the host. And so that got us thinking about the effects of climate and temperature specifically on immune defenses of amphibians, and specifically these defenses that are found in the skin. On the skin of amphibians, there's a variety of things that could be useful against BD, including a variety of symbiotic bacteria that live on the skin, the antimicrobial peptides that I mentioned before that are produced in granular glands and then secreted into the mucus on the skin, and all sorts of things like antibodies and lysozyme. And so one of the goals of our research lately has been to understand how temperature impacts the effectiveness of the defenses that are found in amphibian skin mucus. And we've focused specifically on antifungal bacteria and antimicrobial peptides. So I'm going to talk first about our work on antimicrobial peptides. And so here, my grad student, Matt Roback, did a really complex experiment where we used an injection of norepinephrine to deplete the antimicrobial peptides on frogs' skin. So when you inject them with norepinephrine, they open up their glands and release the antimicrobial peptides. We can wash those off and basically deplete them of that defense. And so we did that to frogs monthly and either held them at 14 or 26 degrees Celsius and either exposed them to BD or did not. And what we found was that in this species, this was southern leopard frogs, there are 4 known antimicrobial peptides. The one we found most commonly is called brevinin 1SA, and that's the one I'm going to talk about because we found it in the most frogs from this experiment. So what we found is that the frogs were able to renew this protection, their antimicrobial peptides, better at higher temperatures. So at 26 degrees, within the month between our injections, almost all the frogs were able to renew the peptide such that we could detect it again. But at 14 degrees Celsius, fewer frogs were able to do that. And then we looked at the amount of peptide that was present. You can see a big difference between the two temperatures in that the frogs at 14 degrees were only able to make small amounts of this peptide, whereas frogs at 26 degrees were able to make much more of it in that month time. So that suggests that temperature really did impact how they were able to synthesize and use this defense. The other thing that we found was interesting was infection with BD itself seemed to impact how much of this antimicrobial peptides they could produce. So frogs that were exposed to BD, this is all at 26 degrees Celsius, the BD exposed frogs renewed their antimicrobial peptides more slowly. So it seems like being infected actually inhibits your ability to produce these defenses also. We did an exposure experiment with these frogs. We found that there was greater mortality in the BD-exposed frogs where they had these depleted antimicrobial peptides, but only at the lower temperature. So there was an interaction with temperature and the effectiveness of these peptides. We did a similar experiment, but looking at the antifungal bacteria on the skin of frogs. And in this case, we manipulated these bacteria on the frogs by putting them through a series of baths that either resulted in a bacterial community that was reduced in bacterial community diversity, one that was intact, and one that was enhanced with a bacterium that we know inhibits the growth of BD. So this is a bacterium called Stenotrophomonas multifila, which we knew from our previous work inhibited BD growth across a variety of temperatures. It also inhibits the growth of many plant and some human fungal pathogens because it degrades chitin, which is found in fungal cell walls. We did not know at the time, and this will become important, that it is naturally resistant to many broad-spectrum antibiotics. So what happened is that this top treatment, our reduced bacterial community, and this bottom treatment ended up very similar in the amount of S. multifilia that they had, because when we got rid of all the competitors up here, pretty much we were left with a high S. multifilia content in 2 of our treatments and a sort of normal bacterial community in the other treatment. So in this experiment, we found that frogs with reduced, like I said, where we tried to reduce their microbial diversity, they ended up with as much of that beneficial microbe as the other group. So I sort of highlighted those as the blue is the ones that have a lot of that beneficial bacterium, and the orange is the one that does not. Temperature impacted the load of BD the load of the infection that these amphibians got, but our bacterial manipulation didn't. So frogs had lots of BD on their skin at 14 degrees Celsius, not very much at 26 degrees Celsius, regardless of treatment. Another thing that we found that was interesting is that at low temperature, frogs with more S. multifilia on their skin, more of that beneficial bacterium, survived longer despite having equal or greater BD loads on their skin. So you can see that here. This is the load of that beneficial bacterium. Our 2 blue treatments have a lot more of that bacterium than the brown treatment, and those frogs survived much longer with BD infections. And in fact, they had as heavy or heavier BD infections than the frogs in the other treatment. So we think that having these beneficial microbes on your skin actually enabled the frogs to tolerate high levels of infection and survive those infections. So taken together, it seems like the effectiveness of amphibian defenses is sort of dependent upon temperature, and that's something that we wanted to look at going forward. And the way we're doing that is now looking across amphibian communities and looking not just at seasonal changes in temperature, but predicted future changes in temperature and how those might impact amphibians. So thinking about how wildlife disease may change in the changing climate, this is sort of the guiding thoughts that we have. So as the climate changes, current pathogens are going to spread and new pathogens are going to emerge. If we want to be able to steward these ecosystems effectively, we need to be able to predict, detect, and respond to disease outbreaks. And to do that, we need to have new tools that clarify the links between climate and infectious disease, predict future outbreaks, and develop disease mitigation strategies. And I sort of stole a lot of this language from part of our contract with the Department of Defense, who's investing in this type of research on a number of wildlife fungal pathogens, including amphibian chytridiomycosis, white nose syndrome in bats, and leptospirosis in island foxes. So one of the ways we've now tried to take what we've learned to make predictions in space and time as to the risk that chytridiomycosis poses to amphibians is using biophysical models. And so these are models where we're downscaling climate data and using models of the physiology of the frog to make predictions of body temperature. And from our experiments, now we have relationships between body temperature and disease risk. And so one thing that we've been able to do now is develop maps like this that we can map the probability that a BD-infected cricket frog might survive to breed during its its breeding season. And so on this map you can see that that probability is much lower in the northern part of its range than it is in the southern part of its range. And one cool aspect of this project that I won't get to time to really elaborate on is that it's informed by data we're collecting from middle schoolers. So we developed a curriculum that teaches about climate change impacts on ecosystems. And as part of this curriculum, the students are making little agar model frogs that have temperature eye button sensors on the inside. They put them out in their schoolyard in different microhabitats. They work with the data and they report the data back to us that tells us amphibian body temperatures in lots of different places and lots of different time periods that we can use to sort of ground check our computer model to make sure that we've got the right predictions for body temperature for amphibians. So it's been a fun way to engage with students and also contribute to our research. So another direction we've been looking at in terms of understanding the impacts of climate change is the potential impacts of climate change on immune development in amphibians. So does developing in a stressful climate lead to frogs with compromised immune defenses? And to ask that question, we've been doing mesocosm experiments at a variety of field stations where we have amphibian larvae developing under different conditions that mimic plausible future climate conditions. So we've looked at drying, early pond drying, we've looked at warming, and we've looked at changes in the community of amphibians in these ponds. So after we have these amphibians develop, we do a variety of immune assays to see whether developing under these different conditions impacted the immune systems of the frogs as froglets and other sort of lasting impacts on the amphibians themselves. And so for the experiment that I'm going to talk to you about, it was a fast drying experiment where we had 3 treatments, no drying, moderate drying, and fast drying. And once the froglets came out of these mesocosms, 5 to 6 weeks later, we did a bunch of different immune function measurements. And when they were a little bit older, about 15 to 16 weeks after metamorphosis, we did BD exposures. So what we found is that survival to metamorphosis was lower in our drying treatments. So a lot more tadpoles made it to metamorphosis when their pond was not drying out. And interestingly, males, but not females, developed faster under the drying conditions. We didn't expect the sex differentiated result there, but that's what we got. When we looked at the bacterial killing ability of the whole blood of frogs about 5 to 6 weeks after metamorphosis, so this is part of their innate immune defense, we found that the bacterial killing ability of the blood increased with larval periods. So frogs that were able to take advantage of a long larval period for development ended up with a better ability to kill bacteria in their blood. Another one of the immune assays that we did was an assay of the swelling response, which is part of the adaptive immune response. And we found that this response was more muted in frogs that had developed in our drying treatments. And so a healthy swelling response is part of a good adaptive immune response. So the fact that these guys didn't have as strong of a swelling response suggests that they're in some sense immunocompromised. And then when we exposed these frogs to BD, we found that frogs that developed in drying treatment showed reduced survival when exposed to BD as juvenile frogs. So that early life larval exposure to these harsh climate conditions was affecting their ability to deal with pathogens at another life stage. And so in this work, like I said, we're looking at other climate stressors, and the end goal of this project, which is a big collaboration with 6 different labs across the country, is to develop models where we can model disease risk under different climate scenarios, but also under different management alternatives to try to figure out if we do have an outbreak, what is the best way to tackle that going forward. And I'm hopeful that this approach will be one that will be useful in thinking about some of these other emerging wildlife fungal pathogens as well. Okay, so now I'm going to dramatically switch gears from talking about my research to talking about a vision for the U of M biological station. And so in thinking about my vision, I first started by thinking about what is the role of a biological field station. And my sense of what is the role of field station comes from my experience in the past 4 years being a field station director, but also having just gone through a major planning process for our field station, where we've been visiting a lot of peer field stations and learning about the amazing things that they're doing, and thinking about the future of ours. And so to sum up what I think the role of a biological field station is in a large part research, being part of a worldwide infrastructure for monitoring and understanding environmental change across scales, from local to regional to global. So I put up here a figure from a publication that just shows the location of biological field stations worldwide. There's many, many of them. And I put this here to highlight that the UMBS, which was started in 1909, it was one of the first biological field stations in this network. So it's been playing a major role in this worldwide infrastructure. So some of the challenges that this global network can address are things like climate change, effects of land use, effects on biodiversity, and how ecosystems and communities are changing over time. Another role I see of research at a field station is to foster collaborations that lead to integrative and innovative approaches to understanding these global environmental concerns. Another role of field stations is to collect, curate, and share environmental data. And then finally, to contribute to informing current and future generations. And I mean by training scientists, but I also mean by communicating science, whether that be in terms of literature, arts, contributing to environmental law, education, policy. In a number of ways, I see a role for field stations research. Under the heading of education, field stations are a great place or one of the key places where we can promote the appreciation of nature, the understanding of environmental processes and human impact on them. So biological field stations ideally are able to reach diverse sectors of society, and students at field stations are often playing a big part in the process of science inquiry, and they do that in an experiential learning setting. So I put up some pictures of students engaging in different activities at research stations, and I thought specifically about the role of graduate training at field stations. It's a place to conduct research, but it's much more than that for graduate students. It's a place to build new skill sets, develop collaborations, receive career-oriented training and mentorship, and to learn how to communicate their science to a broad audience. For undergraduates, it's a place where authentic research experiences have a very long history. It's a place for collaborative and inclusive learning community feel to your education. And it's a place for field studies in the broad sense. So field studies in science, but also field studies in areas such as literature, arts, engineering, architecture, many different fields. In terms of outreach, I see a strong role for community outreach at field stations, both in early education and in engaging community members in understanding the environment and being a part of the science process. So field stations, I see them as a great way for kids to build connections to nature, inspiring awe and interest in understanding in natural processes. And there's a lot of research that shows that if kids do this at a young age, they're going to have this appreciation and connection to nature throughout life. And I think that's super important and a place where field stations can do well. So strengths that I see in the UMBS right now, in terms of research, there's a clear strength in ecosystem ecology, understanding the cycling of chemicals between the biosphere and the atmosphere. Work on climate change there is very strong. Work on freshwater and forest ecology seems very strong. They have some amazing longitudinal data sets that have been collected year after year, are a big asset to that sort of network of global field stations. They have some very unique facilities, a great national and even sometimes international user base, and a great set of immersive field experiences for undergraduates, including RU experiences, authentic research courses, and these new transforming learning courses, which I am completely in love with at this point after hearing more about them. And it's also strong in its interactions with local government and local NGOs. Places where I see opportunities for growth for the field station include building the capacity and infrastructure for year-round education, research, and outreach. So making it more of a year-round field station. attracting new tenure-track faculty to research and teach at the station, removing the financial barrier to help improve diversity, equity, inclusion in summer courses by undergraduates, and the opportunity to develop and encourage more interdisciplinary and collaborative projects at the station. So just looking at U of M and all the different departments that could potentially benefit from research at the station, there's a large number of them that range the gamut from earth and environmental science to art and design to public policy, environmental law. It could be lots of different collaborative engagements up at the field station. I already said that I love the transforming learning model. But also I think broadening in residence opportunities. The station has a great artist in residence program, but I'm thinking that there could be more of these type of community, or these type of opportunities that help us to broaden the community that's up at the station over the summer, maybe in literature or in some aspect of science that's not necessarily always present at the station. And I think we can work toward the built environment at the station becoming a model for sustainability that more is reflective of the research and the education that goes on at the station as well. I think we also need to be working on more K-12 and community outreach that will help us to establish a better connection with the local community at the station. So to sum it all up, my vision for the UMBS going forward is that it will continue to be an internationally renowned field station, having year-round academic community supported by resident and visiting scholars, faculty from U of M who integrate their research at the biostation with teaching and outreach. It'll become an asset to multiple schools at the university. It will be a model for interdisciplinary and integrative research and education, and a model for diversity, equity, and inclusion, and sustainable living. So that would be my vision for the station going forward. And I think that is it, but I am happy to take any questions.
The data you show that the Panamanian frogs are rebounding with some evolution. So they've evolved somehow to tolerate the disease. Are there any negative impacts that you can see from that evolution in terms of the trade-offs and that process has made them, say, weaker at anything?
Yeah, that's a great question. So we know very little about the evolution that has occurred. I mean, basically what we have is the evidence from the antimicrobial peptides that something in there has changed. So I'm really interested in understanding what exactly has changed. That's what we're looking at the exomes and trying to figure out. But it may be that different species have accomplished this in different ways. Maybe they've all done it in the same way. We don't really know. But it's a really interesting question. And I'd be surprised if there weren't trade-offs. I mean, if you think about the extent of selective sweep that happened when this pathogen went through, usually we do find, lots of different changes in these situations. So there probably were trade-offs. One thing I've been thinking about is all these frogs that were taken out of captivity, or taken into captivity and sort of missed this opportunity to evolve with the pathogen for 10 years. Can they now go back? That's probably going to be a bad thing because, they don't have this adaptation and they're sort of swamping out those potentially adaptive traits that have been acquired or been favored over the past 10 years. Yeah, it's a good question. I would love to know the answer over time. Yeah.
This brought back some memories for me because I was living in Panama and my housemate was studying Andrew Crawford.
Yeah.
Studying these, and so I think we remember seeing El Cope before with many frogs, all different colors, and then after with nothing. And my question, that's a comment, my question, so you had this disappearance of the frogs at the community level, and then years later you still didn't see them, and then more years later you do find them. Are these coming back as a result of migration from other places? And geographically, are there areas where the chytrid fungi just didn't hit and then it's being repopulated? Or do you think the other scenario in situ evolution?
Yeah, great question. In some senses, the work that we're doing with Atelopus will help us to answer that question. but my sense is it's probably more in situ. So some of the species that are coming back don't have very large ranges, so they don't have populations that exist in the lowlands where they would have been potentially protected from these epidemics. So some of them are really, you know, mountain endemics that, and we don't think that there are pockets around where BD is not, because we have not found anywhere that BD is not in any of our sampling. So we think it's probably happening mostly in situ, but some of the genetic work that the Atelopus work, and actually Ellie, Bree Rosenblum's student, has worked on a few other species, will help us to look at that from like sort of a landscape genetic context. Yeah. Yeah.
What's your kind of, I guess, more of a perspective question, but what's your long-term opinion, I guess, if you were gonna make a prediction about how in like 50 years we will look at BD? Obviously, you lose some species, we lose some species because of BD, but you know, from another perspective, was this going to be beyond the species that we lose in that sense? Is it kind of going to be a blip where, you know, obviously a lot of species get locked into, like I was looking about the IUCN numbers, for example, you know, 30%, those numbers kind of get, you know, we kind of perceive those numbers, which are reflecting to some degree the like 2004 assessment of what the actual risk is to some of those species. And so I guess I'm just sort of thinking about where you see that headed kind of in the long term. But
yeah, that's a great question. I guess the answer will sort of depend on how these communities continue to change. So it seems like we're on the trajectory where the communities are starting to resemble what they were before. But if you look closely at some of those abundances that I showed, the communities may be coming back in terms of which species are around. You won't get everybody back, but they seem to be shifting in terms of which are the most common species. So it may be that we find that, you know, there are some major lasting changes, even though a lot of the species have returned. And maybe this even affects the ecosystem functioning in some ways. I'm not sure. But yeah, my hope is that it will be more of a blip than anything else, and that certainly was not the thinking for a very long time. But I'm talking to more and more researchers from more and more parts of the globe that are telling me, yeah, we're seeing something similar. Yeah, we're seeing something similar. So it seems in a lot of places we are starting to see recovery, which is great, and I'd love it to be a blip. Yeah, that's my hope. Yeah.
Can you talk a little bit about how you see potential synergies between your role you would have as a director of the biological station and your own research program?
Yeah, so well, I mean, one obvious synergy is that it would be easy for me to do my research at the field station. So the work that I talked about at the end with the mesocosms and looking at sort of taking more of an eco-immunology approach to understanding current and future climate impacts on immune defenses in amphibians is something that I could easily continue doing and expand at the biostation. Beyond that, I mean, my research has a strong, you know, climate sustainability conservation focus to it, and I think that that would bring some strength to the similar focus of the biostation. Yeah.
Switching to more field station administration type question.
Sure.
In your current directorship, have you had any experience with private fundraising?
Private fundraising, you mean in terms of individuals? Yes, yes, lots of experience with donor fundraising, yes. Yeah, so I work closely with the advancement office at the University of Pittsburgh to keep in contact with donors, cultivate donors, have lists of asks, and yeah, right, that's something that I've learned in the past 4.5 years.
Just out of curiosity, do they do the asks there, or do you have to do the asks?
It depends. It's very situationally specific. Depends somewhat on the donors and how they've been cultivated, and but sometimes it's me, and sometimes it's others in the advancement department. Yeah.
So you mentioned talking about diverse equity inclusion initiatives in your current role as director of a station. Can you talk a little bit about efforts you've been engaged in increasing inclusivity, creating more inclusive environments, and overcoming issues?
Yeah, I think the main way we've been trying to do that is to work really hard on the community activities at the station, making sure that when people come up to the field station, they feel like they're included in as a community of academics, and this goes all the way from undergraduates. We actually even work with high schoolers, undergraduates, graduate students, faculty. We have lots of ways for them to get together socially, to interact. We make sure that we have stimulating academic things, like we have seminar series where we invite folks in to give seminars once a week, and we make sure that we have brown bag lunches so that students from all levels can interact with the speakers and sort of feel included in the academic community. We haven't actually had an issue with recruiting a diverse population of students to come up to our field station. So Pitt is a very urban campus, and we have a diverse student body, very diverse student body. And at our field station, the student body at the field station has very much reflected the diversity on campus. It hasn't been a concerted effort on our part to do that. It just has sort of naturally flowed. And I'm not sure exactly what the difference is, but it's something I'd be interested in looking into.
Any additional questions? All right, let's thank Cori.